If you're wondering why Ago has this weird tail here, it's because it's a predicted structure. And so a lot of people have been asking me about like protein structure prediction tools. There was this whole hype about AlphaFold and stuff. So I'll tell you more about that, and also about David Baker at University of Washington. Actually has like this open source method that, when like Google and stuff, they were all secretive about their like DeepMind method or whatever, but he's like committed to making everything open source from the beginning. And so like his stuff actually came out before like the DeepMind paper. So I'll give you links to both of them. And I'm not going to tell you like how much, like how these things actually work, because I don't know all those details and I'm not like a computer science person. But you tend to see like the computer science person and the press and stuff just kind of like freaking out, like, we've solved the problem of protein folding. And then you go and you look at some of these structures and you're like, not really. So it does really well for some structures, but not for others. And it tends to do really well in structures that we have a bunch of structures of already, because it's actually learning from like structures that structural biologists, so like X-ray crystallographers, cryo-EM people, all of those, they go through all this like experiments and stuff to actually determine the structures using these hard methods. And then basically these programs are taking the information from that and from like the sequence, comparing them across different sequences and conservation, and like whether the sequence is like pairwise. So like if this one is positively charged, then the thing over here would have to be like maybe negatively charged, and that sort of thing. So it has all these weird like computer sciencey things, but it's basing that off of what we know based on like all the structural biology work and stuff. And there's still a lot that, like, I think that from my perspective, like what I'm hearing from structural biologists and like people and stuff, is that we're more frustrated that it's being overhyped so much, less frustrated that like we're gonna lose our jobs or anything, because there's still a lot that can only be found experimentally, especially with regards to like proteins that bind things. So like I was showing you with my Argonaut structures, how when it binds to RNA, it like totally changes shape or whatever. But these software predictions, they don't have that capability to understand that. And you might also hear about like people talking like it's going to revolutionize how we make drugs and stuff, because we can like skip years ahead because we already have the structures. Except that in order to do that like drug design type stuff, you need to have like really good level detail. So often when drug design, it's like they're looking for like in like active sites or whatever of proteins. And so you want like really high resolution things, and then they saw the structures of it bound to the compound, and then see where the things are around the compound and stuff, and then make changes based on that, is like the idea with like structure based drug design a lot of the time. So if you only have like a prediction, then you're not going to be able to like see what's really going on. So I'm a little skeptical about that. So I'm not trying to like throw water and stuff. I think there's things that this is great for as a structural biologist. And one of those is like ab initio prediction, for when you have models that are like too low resolution, and there's not a model, there's not a structure of it that was solved before. So with both crystallography and cryo-EM, both, if you have a structure of like some protein or whatever, and then you're solving another structure of that same protein, or it's like a similar protein from a different species, or it's part of the protein, or it's like that protein in a complex or something, you can use the past structures to kind of like help the software that you use to solve the structures, to like help it to like give it a latch on point and say like, so if you have like this blobby thing, and then you have like a little part of the blobby thing, like you could put my palm in the hand and then use that to help orient the like actual physical data that you're getting from your collection method. So whether it's like your electron density map or that sort of thing. So with these methods, you get like these blobby meshy things, and then if you have a previously solved structure, like of a part of it or something, that can help you build the model into the meshy thing, because it gives it like an orientation, like, oh, that's where the like the top or the bottom or the side or whatever, and it helps with that. But if you if you're doing something like ab initio, like from scratch, like there's no previous model, there's nothing you can like dock in there to try to like give it something to latch onto to figure out the structure, that's harder. So one of the great things with like if you have structure prediction or whatever is that you can then use a predicted structure to help you model the rest of it. And it's also really nice to have predicted structures if you're trying to like get something to crystallize or something and you can't, and maybe like there's a region, if you look at like the modeled structure, that it like maybe you can see like, oh, maybe if I cut it off here. Although there's also like prediction tools that you can use already just to like see predicted disorder and that sort of thing. But it also just like gives you a good sense, it can give you an idea of where things might be in space, but it's not like guaranteed or anything by any means. And so I'll show you how you can actually go and look at the predicted structures for proteins. And so I'll show you like in the robetta, or which is like the server thing. I don't know all the things, but it's for Rosetta, which is the open source Baker lab. Seriously, like we need to value this person, this Dr. Moore, because he's done like all this great work for free. I mean, like given it free open access, anyone can do whatever they want with it, and he's been committed to that for like years, and so he's done this amazing work, but you tend to just hear about like AlphaFold because Google, you know. But anyway, so I'll show you how you can submit things to that server, which is cool too, because you can submit like even if you don't like know, like if other people aren't interested in that protein or whatever, you can still submit it, or if it's like that sort of thing. I think you can also do like custom stuff or whatever, but it's all free or whatever. I'll show you how you can submit requests, and you can also see like what's already been done. You can like search for that, and then also the AlphaFold data bank. So the cool thing, one of the cool things about the Rosetta or robetta or whatever, is that it shows you like 5 different models, so you can see like 5 different versions of AGO that it predicted. Sorry, it's my computer's being a little slow. It was here a minute ago. But you have these different models. Yeah, I don't know why my internet's been having problems this morning, but it's not the website, it's my internet. It was really there a minute ago. And it'll give you like the predicted, like how confident they are for every amino acid, so every protein letter and that sort of thing. And you can also download the structures, like download the models and then open them in Pymol, which is what we've been talking about, where you can see like we've been talking about how you can fetch the PDB files of like structures that have actually been solved with like crystallography or that sort of thing. You can also you can't just like fetch it, I don't think, like you normally do, like fetch PDB whatever, but you can download these files and then you can open them in Pymol. You can open them in Pymol. So I've like downloaded the files, and then if you like open, you can then open the file that way, and then it'll show up in your Pymol session, and you can actually even like compare it to like the real structures or whatever if you want to see how well it worked. And so the AlphaFold and the Rosetta use like slightly different methods, so you might see like different results for the two of them. And sometimes they're better at one thing than another. But yeah, so I think it's definitely, I'm not like trying to like throw water on protein structure prediction. I think it's really helpful. I just don't think we can say like the protein problem has been solved, folding problem has been solved or whatever, because, well, it was solved by like biochemistry. I mean, the molecules solved it pretty well. But in terms of like us being able to like totally predict protein structures, often like the most interesting regions of a protein are the parts that are not going to be predicted well, because if we knew exactly what it was like, then all molecules would just be like super similar, and that'd be not fun. So yeah, and also the things with like binding and changing shape and all of that stuff that you'd miss out. Okay, but anyway, so let me take you to the computer and I'll show you how I can do this. Okay, so I got the I got my internet working. It was the internet, I swear. It wasn't like this site. So you can see, yeah, there's some regions where it's just doesn't really know what to do. Like this beginning region. Often you'll find that the starts and ends of molecules are like the hardest, like are usually pretty flexible and stuff, so they'd be hard to predict. But yeah, so you can see it gives you different models. So there's like 5 different ones. And for each model, so right now it's just colored like rainbow, so it's like starts with one color at like the N terminus, and then it goes all the way through and then to the C terminus in red or whatever. So basically the N terminus is like the start of a protein, and the C terminus is the end. um end and But you can also, yeah, so there's like, it'll give you 5 models. On the right over here, what you're looking at is, it's like angstrom error estimate for each like position. So an angstrom is like a measure of distance, like a really small measured distance that we use in structural biology. And so it's like saying like how far from where it would, like how uncertain are we about the position of these specific amino acids. So it's like showing you like, so AGO has 859 residues, and so like for each one, like how confident are they in the prediction or whatever. So you can see at like the ends and stuff, there's regions where it's like we don't really know. So if you want to color it, you can color it by like the error estimate. And now you'll see like that region, like that long tail thing. Oh, see, now it's like curled up into the protein, which is very, I doubt this structure, but that's a very interesting curly loop. But you can see that the regions that they're unsure of, and so like in the different models, they're going to be in like different conformations or different shapes or whatever, depending on how confident they are. So yeah. So this is what you see in the, I'll show you how I got this all in a minute. And then you can download it from here. So this is Robetta. So this is the Baker lab. And so David Baker at the University of Washington in Seattle, at this the UW Institute for Protein Design. So they've done a ton of great stuff. And so this was their recent paper that came out in Science about their work. And so you can submit to this like robetta server. And so they have a bunch of FAQ and all sorts of stuff. But yeah, if you go to project, you can like find out more stuff. But if you go to like structure prediction, so you can submit. So you have to like register, but I mean, this is all like free and open access and stuff. But I haven't actually submitted things because things I've wanted are already in this queue. So you can see these are like some requests that people have made. So apparently She Crazy really wants to know the structure of a lot of different things. But I think they say it should take like 1 to 3 days or something. You can also look at some example results. So if you're wondering what these things are, these are like the structure, like prediction, like the secondary structure prediction. So like whether some things like a helix, so like an H would be like a helix, and that's predicted based with these like different softwares. And so you can see which regions that are predicted to be different things by like these other softwares that it's I think it takes those into account, but I'm not exactly sure where it's getting all of its stuff. But you can also search the queue. So like I searched for AGO 2, which is what I was interested in, and so I found that someone had like wanted it already, and so I looked into that. I haven't actually tried, like, so if you search for sequence, I'm not sure if you can like search the actual sequence. I'm not sure how that all works, honestly. But you can, yeah, see predicted protein structures. So like try searching for something, and if it's not there, then you can always submit it to be solved, or at least to give you some predictions. So that's the robetta. And then when you download models, then you can then go into Primal, and you'll see the model here, and then you can play around with it and do everything just like you would if it were like a structure that you got from the PDB, and so you can even like compare it to the PDB and that sort of thing. And so yeah, so more on the PDB, more on Primal, all of that different stuff in the previous videos. I'm not going to go into that today. I need to get back to my work soon, but I was wanting to see what they were doing with Ego, and so because I was already working with Ego and Primal, but with like crystal structures. But anyway, okay, so the one you so I highly encourage you to like tell people about David Baker and all his work, and like how really he pushed the field and everything is open access and it's all really great. But you don't hear about it as much. What you hear about more is this like AlphaFold. So this was the Nature paper in AlphaFold, and what they so this is like Google, DeepMind, those people. So basically they had solved, there was like this protein folding competition thing or whatever, and they did like amazingly well, and then, but they didn't like publish like how they did it or anything. And so then the Baker lab, they got like, they tried to kind of like see if they could reproduce the AlphaFold type stuff. I'm not sure if they did it, like if they were actually trying to do the same thing or if they were just like trying to find a better way, because they use slightly different ways. But in the Baker lab, then like put everything like totally public, and then AlphaFold went public with their stuff too. But so basically, there's this like AlphaFold protein structure database. So this is a nice thing about AlphaFold, is that it has this like, there's this huge database, and I'm not sure if like, yeah, I don't think that they let you I don't think they let you like submit custom things or whatever like the Rosetta does, but they do have a lot of structures already here. So like if I search for Ego 2, yeah, so they have it for like different species and that sort of thing if I just want the human, or you could put in like the uniprot accession or that sort of thing. So like if you go to uniprot, if you're not familiar with that, I don't think I've really talked about it much, but it's this like protein database with a whole bunch of information about all different things. And so you can like align structures and stuff. It's really great. So this is like Ego 2 human, and so you can see that you have all this information here. It links you to structures and all that sort of stuff, but it has this uniprot like code, like QN whatever, so I could also stick that into here and it would find it, or I could stick Ego2_human, but you can also just like search for whatever protein, like if you don't know the code and stuff, that's fine. But also, yeah, uniprot is a really great resource. I'll probably hopefully do a post on it in the future. But when you go to one of these entries, so you can like PDB file or MMSIF file, those are two different like structure file format things or whatever. So either one will be able to open in Primal. And so this is just showing you the sequence, and then for each it's giving like a confidence score and shading it by the confidence score. So you can see like the blue regions it's really sure about, and the orangey regions it really isn't as sure about. And then this is just like more error stuff, predicted error, whatever. I'm not exactly sure. It'll tell you all about how they predict that error and stuff or whatever. But yeah, so they also have this, it's like hooked up to, I think it's connected to like the MBL EBI, which is also like, I don't know, I think they're all somehow, they're connected to like further information about the protein. So like the gene name and the structure, the function, and that sort of thing. So yeah, so see, it's directing you to UniProt, which is where we just were, and it can also direct you to the PDB, so the PDB, remember, is like the protein data bank, and that's where all of like the crystal structures and that sort of thing are. This is like the PDB EKB. This is like, so this is the European version. I've mostly been showing you like the RCSB, so like the United States one that's like based out of Rutgers. But this one's like the European data bank, but they have it like hooked up to like further information. But yeah, so PDBs, like you can download structures of the different proteins. And so yeah, so the PDB, so I use like, this is the version that I showed you most. And so yeah, so 4F3T, that's the Argonaut structure that I was using before. So you can see, you can find it here. And yeah, so that is what is so that's basically how you can play with these things. So yeah, so you can then you would be able to open the AlphaFold predicted structure here as well, and you can compare them all. You can color them different ways. You can play around. You can do that for like any of the proteins that they have. And remember that with the Robetta, so with the Baker Labs software, it's based on their like Rosetta. I think Robetta is their server, and then Rosetta is like the software that it uses. But yeah, so you can submit requests for them to do whatever. And yeah, so I highly recommend you to check it out. And to don't forget about the Baker Lab, that really deserves a lot more credit and attention. So it makes me mad when I only hear about AlphaFold. But yeah, they're all doing really amazing work. It's just, so I don't want to be like, sound like I'm discounting them or anything. Like, they're all really, this is like really amazing work, and I'm really happy about it. I just don't want it, like, I just don't want people to think that we now know the structure of every single protein, like all of them in all their regions and every species and everything. Like, I don't think, like, I hate hearing like the protein folding problem is solved. Like, I would not say that. And I don't know if it could like ever really be solved, whatever solved means or whatever, but there's just so many. Biochemistry is always like one step ahead of everyone. It's biochemistry is really just amazing, and things do things that you wouldn't expect, and there's always new things to learn, and so yeah, so I think this will be a really powerful tool to help structural biologists. So like, and there's a ton of things it's good for, like I was talking about with like the ab initio, like modeling and that sort of thing. But yeah, and so sorry I used like a bunch of terminology and stuff without really explaining things as much as I usually do. This was just kind of a bit of a rant, fueled by when I was just like playing around for my work and that sort of thing, and hearing people say things. And people have been asking me about it, so I thought I'd make this little post, and now I need to go back to working in Primal for making figures for a presentation. So yeah, so hope that this helped you understand a little of what was going on. And sorry I don't like have all the computery details, but I'll give you the links to those sites and papers, and yeah. I realized I forgot to mention one of the really great things about these protein prediction tools is that they can actually like help us figure out what questions we want to ask with like biochemistry experiments or whatever. So sometimes there are things that you can't learn directly from a structure, like whether a specific region actually does something, but you can see, oh, it looks like it might do something. Let's test if it actually does that something. So then we can use techniques like site-directed mutagenesis, where we can like make specific amino acid changes, so changes to specific protein letters, and then see if the changes are what we would expect. And so we could also do this sort of thing to try to like see if structures are like the modeled structures are likely correct and stuff, because if you test different, basically like based on how they interact or whatever, if you change them, then they should change the things or whatever. But basically, there are things that we would be able to get from a model, even if it's not totally accurate, that we can then allow it, like give us ideas and theories that we can test with like biochemistry experiments. So like seeing if things bind, seeing if things like catalyze reactions, like so speed up reactions, various reactions, like, oh, that looks like, or maybe you see like, oh, that looks like something that I saw this one time and whatever, this other protein. But there are also other tools and stuff that you can use. So this, all of these advances and stuff, they're building upon years and years of work. So there's been like structure prediction tool, like disorder prediction, like secondary structure prediction, comparisons between protein structures, so like domains, like parts of proteins that kind of like fold separately, or they have this whatever, they have like a special fold or whatever. So if that's in one protein, then and you see that sequence and you think, oh, that probably folds that same way. So there's different tools, and all these, like these softwares and stuff, they build upon all of that knowledge that's been built up over the years. And so it's not just like the structure that they take into account. It's like all of these years of modeling based on the structures. So it's like, it's not like it's a huge advance, yes, but it's not like it's just like coming out of nowhere. But yeah, so it's a complementary technique to structural biology, to biochemistry, to all of these things. So I think it's a real win-win, as long as it's used correctly and not just like hyped about and overconfidencized or whatever. Yeah, I make up words and I'm tired, but yeah. So I just rambled a lot, and I will let you guys go now.